Evening, everyone. Hello, all. Hope I'm audible. So, today we all have joined to yet another session of Biorix. Starting with what exactly Biorix is. Biorix is a student run biology community built for curious minds across the life sciences, from genetics and microbiology to ecology, zoology, botany, and conservation. It serves as a collaborative platform where students connect, exchange ideas, and engage in meaningful scientific discussions. So, as a part of our exploration of the wonders around us, today's session focuses on arachnids. Today's session is titled Beyond Spiders, the Hidden World of Neglected Arachnids, with insights from Pseudoscorpions. To lead the session with us, we have Dr. Jithin Johnson. To speak a few words about Dr. Jithin, he is a specialized arachnologist and assistant professor at Bharat Mata College, whose work focuses on the evolution, biodiversity, and systematics of pseudoscorpions in the Western Ghats. His research has led to the description of several new species. Molecular phylogenetics is also his area of expertise to genetically analyze cryptic species. His publications in molecular phylogenetics and evolution argues that Western Ghats serves as a museum of ancient biodiversity for Indian pseudoscorpions. We are very delighted to have him with us today and look forward to an interesting session. Let us all warmly welcome Dr. Jithin. Sir, over to you. Sir, you're not audible.
Can you hear me right now?
Yes.
Okay, thank you, Gayatri, for the warm welcome. So I'll just get on to the talk. I'll just present my screen. Just give me a moment. All right. So I hope you can see my screen and I'm audible, right?
Yes, sir.
Okay. So today I would like to enlighten you to a new hidden world of arachnids that is beyond spiders, especially with insights from certain animals that I've been working on for the past few years, that's pseudoscorpions. So before getting into the arachnids, I would like to bring your perspective our perspective onto biodiversity studies, that how we look into the biodiversity studies. Conventionally, we looked into big charismatic megafauna to understand biodiversity. And the problem or the limitation of this is that these large animals, like the large mammals, like elephants, tigers, they have a very small evolutionary story to tell us, because most of them have diversified post Cretaceous or in the Eocene period. However, if you look into the microfauna, that's the most microscopic invertebrates, like certain nematodes or certain insects or certain arachnids. That's usually we find them on the soil or under the soil or in the leaf litter, and these have a very ancient diversification spanning, like they have like origins from the Jurassic or even the Triassic, and they have a large evolutionary signal that they can convey to us. And this is my primary interest, why I am interested in such kind of microfauna, or especially arachnids. So just introduction to the perspective of how we look into biodiversity. And coming to Arachnida, so Arachnida is phylum, it comes under the phylum Arthropoda, and the flame arthropoda is coming under the subclass Chelicerata, and it consists of four groups, which is the sea scorpions, that is the Europterida, that's a fossil or the fossil group that originated in the oceans. And then we have the Pycnogonida, that's the sea spiders. And then we have the Scyphosura or the horseshoe crabs. And the most relative to the horseshoe crabs is our class Arachnida, which consists of the spiders and the other eight-legged creatures. So coming to the body plan, they have a prosoma and a opistosoma that consist that's commonly called as the cephalothorax and the abdomen. And a unique feature of the Chelicerata, or the class subphylum Chelicerata, is that they have six pairs of appendages, of which the first pair of appendage is the mouth part, which is called as the chelicerae, and hence the name. And the second pair of appendages is chelate pedipalp that they use for subduing their prey. And then they have four pair of walking legs. And what differentiate this group from the other arthropods like crustaceans or insects or say millipedes or the myriapods is that they don't have any antennae. And instead of antennae, these chelicerates, especially arachnids, have certain sensory hairs that's called trichobothria that helps them sense their environment. Now, this is the broad classification of the class Arachnida. So class Arachnida has 16 orders, of which four orders that you can see on the right-hand side are extinct. And there are about 12 extant order, of which the most common ones are like spiders, scorpions, and you can also see acariforms and parasitiforms, that's the ticks and mites, that's very common. And beyond that, most of us might not have we might not be familiar with the other arachnid orders. So I just want to try to enlighten you with this and get some kind of information through this talk. So if you look into the difference between the common spiders and scorpions, we have the venom in the chelicerae, that's the mouth part, and these are also called as the fangs. Whereas in scorpions, these venoms are located in the elongated metasoma, or the tail that we call it as, and it's the stinger at the end of the tail that has the venom. And if you look into pseudoscorpion, that is very similar to scorpions if you look into the appearance, but they don't have the tail or the elongated metasoma. The venom here is located in the pedipalps, or the first pair, the second pair of chelate appendages. And in the case of spiders, you know that silk is produced in their abdomens in the organ called as spinnerets. Scorpions don't produce naturally any silk, but pseudoscorpions, they also produce silk, but not as vastly or as dense as spiders. They don't build it for creating webs. Instead, they use it as a kind of a refuge system during their molting time or during egg laying period. So these silks are produced in glands that comes out of their mouth part, or the chelicerae. So this is a basic difference between spiders, scorpions, and the pseudoscorpions to start with. Now, if you look into the classification of arachnids, we have the mega diverse orders, that is the spiders or the araneae, and the acariforms and the parasitiforms together. We call them as ticks and mites. These are uh, coming in the very high diversity, like we can see tens of thousands of species. For example, spiders, as of now, we have 53,000 species, and about acariforms and parasitiforms together, we have about 58,000 species. And then we have the mesodiverse order, whose species number is medium range, that is in the numbers of thousands. And we have the opilions, or the harvestmen, and the camel spiders, or the solifuges, the scorpions, and the pseudoscorpions that come under this particular order. And I'll just briefly introduce you to the mesodiverse orders.
We are not able to see the presentation.
Oh, sorry.
Screen is not coming.
Okay. Is it?
Sir, I think it's a personal issue. We were able to view it.
Is it visible now?
Oh, no.
Okay, I'll just share my screen once again. Okay, is the screen visible right now?
Yeah,
Yes,
it's
sir.
visible, and it was visible earlier also. It was a problem with
All right,
one
no
of the
problem.
viewers.
Now
Okay, no
it
problem.
is okay.
Thanks for letting me know. Okay, so coming to one of the groups called as the opilions or the harvestmen or the daddy long legs. These resemble the faucet spiders that or the cobweb spiders that we see inside our houses, making cobwebs. But these are not spiders actually. These are another group of arachnids called as harvestmen, and they have a globose body and they have a fused prosoma and opistosoma, and they don't have any venom glands or silk glands. And one of the unique features is that they have certain defensive scent glands called as the oesopores. And these oesopores are similar to what we find in millipedes that produce hydrogen cyanide when they are getting irritated. So, yeah. And they also show certain kind of gregarious or community aggregation that they do it for getting humidity or to avoid predators. And they also sometimes can purposefully uh, lose their legs in the process called as autotomy and show twisting reflexes. So they can be seen like in a waving motion always with their legs, and that's a twisting reflex that they usually show in the habitat. Now, another group called as the camel spiders or the sun spiders or the wind spiders, they are having enormously large chelicera. They are also very rapid in their locomotion, and hence they are called as the wind scorpions. And they are mostly nocturnal in nature and hence called as the sun scorpions. And so they try to avoid the sun during the daytime, and usually they are active during the nighttime. They have sensory flagellae on the chelicerae. So that's a flagellae is a kind of a sensory organ that they have in their mouth part that helps in sensing the environment. And mostly they are adapted to arid or desert habitats. Again, they don't have any silk glands or venom glands. Now, coming to the scorpions that we all know, like they have a segmented metasoma, which ends in a chelson, that is the stinger tail. And then they have raptorial pedipalps that they use it for prey capture. And they also have this cephalothorax or the integument that is able to fluoresce under UV light. And they also produce this venom that has a wide cocktail of peptides that is of medical importance. So another behavior of scorpions is their mating dance that is called as the promenade ado, that they perform, that the male captures the female and brings her to a particular safe, sweet spot so that the spermatophore can be transferred into the female genitalia. And another interesting feature of scorpions is that they show extended maternal care. That is, the mother carries the young scorpionlings on the abdomen until they get mature enough. And the venom is different cocktail. It's a mixture that varies upon different species, and this is an evolutionary diversification with respect to the different prey specialization. Now, coming to the micro diverse orders, so these are the most least diverse species coming under the arachnids. We have the Amblypygi, that is the whip spiders, or the telephonids, or the whip scorpions, the schizomids, the palpigradi, and the rhizinulae. So coming to the whip spiders, otherwise called as the tailless whip scorpions, these are again non-venomous and nocturnal. You can see them under rocks or in caves, and these have flattened body and elongated raptorial pedipalps. And also their first pair of legs is acting like a whip that is sensory in nature. And they also show this kind of maternal care, just like what scorpions show. And coming to Europygi or Telephonida, or otherwise called as vinagrunes, because they spray vinegar or acetic acid as a chemical defense. And they have this whip-like telson, like the tail, that's elongated. And they also have a long flagellum. So if you remember, flagellum is a structure that's found in the chelicerae, that is the mouth part that helps them in sensing the environment. And they also have a very robust and um, strong pedipalp. Now, coming to schizomids or the short-tailed whip scorpions, they are leaf litter specialists. usually found in arid environments, and they also show a kind of courtship behavior, which is called as a vibrational courtship. They are also having a very rapid ant-like movement and have a prosomal hood that is uh, hinged and arched cephalothorax that covers the chelicera. And coming to microwhip scorpions, these are very tiny creatures, like less than 1 millimeter in size, very common in leaf litter. They have a whip-like flagellate telson. They don't have any venom glands, fossil glands. Again, they are very humidity dependent and can act as very niche indicators. Um, Now, coming to hooded tick spiders, these are rare arachnids, which has certain ventral adhesive organs that help them in uh, getting attached to the substrate. And they have something called as the hinged cuculus. Cuculus is the hood-like structure that is seen covered over the chelicerae that they can retract and move accordingly and thereby expose their chelicerae. So that is why they are called as hooded tick spiders, and they also look like small ticks. And these are again eyeless, and they are mostly found in leaf litter habitats or cave habitats. Now, this is a comparative morphology of the non-spider arachnid orders that I put into. Like, I'm not going into details. If you want more details on the lesser arachnid orders, there is a World Arachnid Catalog where you can get into the details of different arachnid orders. And now coming to my interest organism, that is the pseudoscorpion. The main problem with this pseudoscorpion is the size. And if you look into this picture, you can see that there's a small pseudoscorpion just next to the spider over there. So there it is. So pseudoscorpions are very small organisms. You can see their pictures. This is the normal size that we see them. Like we get them around 2 to 3 millimeter average size. And you can find them in different habitats. And first of all, Aristotle has already described pseudoscorpions in 350 BC in his book Historia Animalium. They are dorsoventrally flattened arachnids, and they have a lifespan about 2 to 4 years. The pseudoscorpions were also previously called as Chilonithida or Chilonati. So the order was named as Chilonati or Chilonithida because they have this um, chelate appendages. So if you look into the distribution of pseudoscorpions, we have about more than 4,300 species distributed worldwide in about 26 extant families and one fossil family. And in India, we have about 160 species distributed in 17 families. Now, if you look into the habitat where we can find the pseudoscorpions, you can find them everywhere, starting from dry deserts. You can find them in intertidal zones where they live submerged. They can tolerate submerged under water during the tide period. They can also be found under snow-laden soil in the Arctics. Except for Antarctica, they can be seen in every other continent. And they are mostly in our forest or in our surroundings. They can be seen under leaf litter, in tree hollows or barks, under the rocks or in the soil, or even they can hitchhike in certain flying insects or certain other animals like birds or mammals or bats, and they just use them as a passive dispersal method for getting from one place to another. Now, this behavior is called as phorecy. So where they passively disperse from one place to another because they themselves cannot fly or they themselves cannot move very rapidly. And this behavior has evolved as a result of a community feeding and a hunting strategy. So many pseudoscorpions come together to feed on a larger prey. And at that course, the primary objective is to hunt down the prey. But at that time period, when the prey just flies to another place, they get a possibility of establishing a new colony elsewhere. So that's a passive dispersal mechanism called as phorecy. Now, another interesting behavior that pseudoscorpions show is that the social parasitism or the cuckoo behavior. So just like a cuckoo or a quail or like the bird that lays eggs on the crow's nest and makes the crow take care of the young ones of the cuckoo. Just like that, here we have a cuckoo behavior or the social parasitism between Paracernus melanopicus species and a Paratemnoids nidificator. These two species belong to two different families of pseudoscorpions. But the interesting thing is Paratemnus nidificator uh, or Paracernus melanopicus can undergo chemical camouflage, and they can lay egg in the nest of Paratemnus nidificator, and then the later will take undergo a parental care of the nymphs of the former one. So this is a kind of paternal care that they do, and that's interesting. And this is found in about 1 to 2 families of pseudoscorpions, not in all pseudoscorpion families, and mostly the interesting behaviors that I'm going to tell about pseudoscorpions. These are most derived families exhibit these kind of behavior. That is the primitive pseudoscorpions, or those pseudoscorpions that have originated earlier on, like very in olden times. They don't show these kind of complicated behaviors. They don't have any silk glands. They don't have any venom. But most of the recently derived species, or the diversified species, or the diversified families, they show these kind of complex behaviors. Now, another thing is matrophagy. So that is when there is food deprivation. In order to prevent cannibalism among the nymphs, the mother will sacrifice herself to the nymphs so that they get enough food. And so, yeah, this is another social behavior, or paternal maternal behavior that has aroused in different pseudoscorpion families, most of the recently diversified pseudoscorpion families. Another behavior that is very similar to the scorpions is that some of the pseudoscorpion families also uh, exhibit mating dances, where you can see the male is kind of they lay a spermatophore on the surface, and then they capture the female and drag them towards the spermatophore so that it is taken up by the genital opening of the female. So during this course of time, there is a kind of a ritual courtship dance. uh, Again, that is very specific to different species and mostly present in derived or recently diversified families. Now, coming to the morphology, we have a prosoma and a metasoma, or the opistosoma, that is the abdomen, and a pedipalp. And what makes it different from scorpions is that they don't have the median eyes of the scorpions. Instead, they have lateral eyes. And also, they don't have the tail of the scorpions, or the elongated metasoma with a single tail. So that's how scorpions and pseudoscorpions differ. And also, pseudoscorpions are very tiny in size. Otherwise, more or less, they are very similar in their behavior or in their morphology. Now, another important structure as far as pseudoscorpion is concerned is that the venom apparatus in most of the earlier derived families, there is no venom apparatus, and and if there is venom apparatus present, they can be present in either one of the fingers or both of the fingers. And depending upon that, we have a different family classification. And also, they can produce silk, and the silk is not for web construction, and it is just for a protective architecture. As I told you, it is just a refuge kind of structure that they produce during molting period and during their egg laying period, when they are most vulnerable. And some of the silk glands, mostly they are found in the carapace region, and they open into the chelicera, or the mouth part, and sometimes they extend deep into the abdomen as well. Now, if you look into the status of pseudoscorpion research from India, we have mostly foreigners who have worked on the pseudoscorpions from the 1930s or the 1940s. We have Joseph Conrad Chamberlain from US and the Max Bayer from Germany, who had worked on extensively on pseudoscorpions and produced monographs. And if you look into the Indian pseudoscorpion works, we have the monograph by Murthy and Anandakrishnan in 1977. That is one of the most extensive monograph work on pseudoscorpions, mostly focusing on the Himalayan region and the Madras region. But post this work, there has been no taxonomic works carried out in this particular group. So there has been a very large gap of pseudoscorpion studies compared to what has been happening in the world. For example, if you look into the global scenario, in 1778, pseudoscorpion was first described by the entomologist De Geer, and then in Chamberlain and Bayer had produced the first classification system in 1930s, and by 2000s we had Mark Harvey and colleagues who were looking into their biogeography and distribution patterns. And currently we have many colleagues working on phylogenomics and proteomics and venom studies and silk studies. So more kind of studies are going on all over the world, except we have a large gap in India. So that's what interested me into coming into this particular group. Now, another question was where did these pseudoscorpions evolve? So if you look into the structure of pseudoscorpion and a solifuge and a scorpion, they look very similar. Like the pedipalps of scorpions and pseudoscorpions are similar. The solifuge have a very robust chelicerae. Pseudoscorpions also have them. So there has been a doubt where to place these pseudoscorpions and what are their sister relationship with other arachnids. There have been a ton of analysis, like sometimes they have been placed together with acariforms, that's the ticks and mites. Sometimes they have been placed with solifugae, that is the sun spiders, and sometimes with the Arachnidae or the other groups. And recently, in 2021, this higher level sister relationship was resolved by a group of colleagues, and they have placed scorpions as sister to pseudoscorpions in a clade called as panscorpions. Now that makes sense because scorpions and pseudoscorpions are very similar in their behavior or in their morphology, and now we can say the molecular data also hints towards the same thing. So this is based upon transcriptomic studies and different other multiple independent evidences, lineage of evidences. Now, these pseudoscorpions, they have been here for they originated some 400 million years back when there was this Panthelesic Ocean, when there was still Pangaea land, or there was no Gondwana land and Laurasia, when there was, so that was the time when pseudoscorpions originated in the um, Pangaea land. That's what we estimate. And the oldest record of pseudoscorpion is from the Upper Devonian, that is 392 million years back. That's from Gilboa deposits in New York. And if you look into the right-hand side, you can see the present-day classification of pseudoscorpions into different families. And the different families of which the basal pseudoscorpions do not have any venom glands or the silk glands, whereas the recently derived pseudoscorpions, which comes under the superfamily Cheriferoideae, that is having 4 families, Carnitidae, Califeridae, Atemnidae, and Vithidae. These are the groups that show the parental care, foracy behavior, or they have venom apparatus, and they have courtship behavior. So this is the classification of pseudoscorpions. And now coming back to fossils, so we have a fossil from the Cretaceous that we modeled, and this is kind of a 3D model that we obtained from micro-CT scan that helps us in understanding the different structures of fossils. And why I wanted to show you this is that, so we have on the left-hand side a present-day pseudoscorpion that is very similar to the fossil pseudoscorpion that you see on the right-hand side. It is like there are no apparent differences that you can actually make out of these 2 different specimens. And this is because these specimens have not undergone any phenotypic or morphological changes over millions of years because they are very adapted to their environment. They just don't change their body plan. And this phenomenon is called as morphological stasis. And that's why these organisms are called as living fossils, because they are very old and yet they retain their primitive characters or their body plan as such. Now, some questions that I am interested in while looking into pseudoscorpions is to understand biogeography, that is how biodiversity is distributed through space and time. If you look into this Alfred Russel Wallace map, he has divided the world into different biogeographical realms depending upon the diversity and endemism and different composition of the flora and fauna. So mostly this is the composition of the fauna. And so we have our, in the Oriental realm, we have our Indian subcontinent and the Western Ghats and the Himalayas as different biodiversity hotspots, which is rich in different biodiversity, and also endemic animals are present. So I just want to use these pseudoscorpions as different, as a biogeographic tool to understand their patterns. Now, India is very special because India has different, it is a kind of a isolated island. Once it was part of the Gondwana land, and then once it drifted out from the Gondwana, split it from Africa, and then it was moving towards the Eurasian plate. It was an isolated island for about 30 to 40 million years. And also once it collided with the Asian plate, resulting in the formation of Himalayas. Now we have in the northern region, we have the mountainous Himalayas. Towards the northwest region, we have the arid deserts. And towards the south side, we have the peninsular India that is covered by ocean in all three sides. So even now, it is bioclimatically, it is can be considered as a island. So there is a lot of endemism and diversification or in situ diversification happening in India, and that makes India very important area to study such kind of patterns. So what the important questions that I was interested in asking was, what is the evolutionary origin of Western Ghats fauna, especially Western Ghats pseudoscorpion fauna, whether they came from Gondwana land, that is, they came from Africa, or they had relatives in Australia, or they were recent dispersals from Asia after they collided, after India collided with the Eurasian plate. So I just wanted to check whether they are ancient lineages or recent dispersals. And also I wanted to check the role of vicariants or certain geographic gaps and how they influence the distribution of the Western Ghats fauna, or especially pseudoscorpion fauna. And also how certain processes like geoclimatic process and ecological processes like the Deccan volcanism and the onset of the monsoon climate has influenced the diversification of Western Ghats fauna. And for this, so I chose a litter-dwelling pseudoscorpion that's called coming under the tribe Tyrannocothonini, and they have very specific microhabitat preferences and poor dispersal capacity and show short-range distribution. And I sampled across the Western Ghats, especially across the Palghat Gap, because I wanted to check how they dispersed across the Palghat Gap. And then a combination of methods were used for sampling them, using some garden soil sieve, or using a Winkler's sifter or a Winkler's extractor, or using manual search, or using a method of active extraction of soil fauna that is called as the Berlesey-Terugren funnel. And this is the global distribution of Tyrannocothonini across the globe. You can see that they are mostly distributed towards the tropics and subtropics. And to get a representative sampling, I also obtained DNA data from different parts of the world. And my results actually showed that the Western Ghats fauna, or the Western Ghats Tyrannocothonini, are very ancient relics, having roots to the Gondwana period. That is, they diversified during the late Triassic in the 234 million years back. And most of the pseudoscorpions, they had their sister relatives in Africa. So that was one of the hypotheses, or one of the answers to the hypothesis. And second one is regarding the nature of pseudoscorpion fauna in the Western Ghats. So if you look into the different geological layers and the climatic as well as the molecular data, you can see that all these combined together give information about a deep time stasis, that is the evolutionary signal. And since these organisms cannot disperse over a long period, they are very efficient indicators of um, uh, climate change or habitat disturbances. And what we obtained in this study is that the Pseudoscorpions from Western Ghats were uh, occupying regions of the Western Ghats there that acted as a refuge, and that acted as a refuge during different climatic as well as geological changes, like for example, the Western Ghats orogenesis, or the onset of monsoon, or the Deccan volcanism. During these kind of bioclimatic processes, certain regions of the Western Ghats acted as a refuge for these ancient species. And hence we can call that Western Ghats is acting as a living museum where there are a lot of ancient species. And this is not a pattern that we see just in Pseudoscorpions. We can see also them in different other arthropod groups. So before coming to that, I'll just briefly discuss what we have here. So I also obtained that the Palgat gap, which usually shows a kind of a vicariant barrier. It is naturally acting as a vicariant barrier because it is 40-kilometer wide, and we have genetic structuring of organisms on either side of the gap. But in the case of pseudoscorpions, we obtained that it is not an absolute barrier, but a filter which allowed dispersals during different periods of time. So you can see that this, what is represented here as D1 to D6, is 6 events of dispersal that allowed pseudoscorpions to traverse through or across the Palgat gap. So it is Palgat gap was a filter, not an absolute barrier. And also we can see that many pseudoscorpions had an ancestral origin in the low elevation forest. And then later on, when there was sufficient environmental factors or sufficient niches available, they occupied high elevation montane forest. So they had an ancestral origin in lowland forest, and later they dispersed into highland forest. So this pattern is also very common. You can see them in many fossorial animals. So you can see them in pseudoscorpions, you can see them in scorpions, you can see them in blind snakes, you can see them in beetles, centipedes, Sicilians, and also certain vertebrates like the Nasica batrachidae frogs, which are again fossorial. So most of the fossorial animals are actually protected, or they kind of get a refuge from the different bioclimatic change processes that happens outside in their environment, and they get protective refuge in the soil ecosystem where they are living. So this is something that is interesting about the arachnids, or the lesser known arachnids, especially pseudoscorpions. Now, there are again a lot of other things to talk about pseudoscorpions, because we have pseudoscorpion venom research that has been in the stage of infancy. We have studies from the 2021 that have the first transcriptomic studies of venom from a pseudoscorpion species that has a lot of neuropeptides and a cocktail of proteins that can be of medical importance. And so venom research is in the infancy stage, but as we know, in the case of centipede venom or scorpion venom or spider venom, as they have many medical importances, pseudoscorpion venom is also getting into that track. So that's an emerging field of research that is of something of interest. And another thing is about sustainable agriculture. We can see that the pseudoscorpions, they live in harmony with honeybees in honeybee hives. So in honeybee hives, there is something called as the varroa mite that feeds on the honeybee and reduces their fecundity. So these varroa mites are actually enemies of honeybees, and pseudoscorpions actively feed on these varroa mites, and hence they help these honeybees, and the honeybees are left unharmed by the pseudoscorpions. So what when we try to understand the ecology of the species, we can build something like a hive, which is having a base structure where we try to grow pseudoscorpions so that the pseudoscorpions can grow and they can move upwards towards the beehive and feed on the varroa mites. And whenever they want, they can just go back to their enclosures at the base of the hive. So this is a kind of a prototype honeybee hive that was recently constructed. And you can also see that pseudoscorpions are available in the internet, sold uh, in the European countries for this purpose. So sustainable apiculture is something that can be looked into uh, while using pseudoscorpions as biological control agents. So, but understanding their microecology is very important. And as I mentioned before, there is a huge gap. We right now most of the studies are focusing on systematics and molecular systematics and phylogenetics, whereas ecology and behavior studies are very lacking. It's not only in India, but globally, it's very scarce. So, what I want to put forward to you is that we can join the next wave of integrative taxonomy, where we look into different species, not only describing new species, but looking into their biogeography, how they uh, are distributed, how they evolved. So, looking into their evolution and also looking into their behavior and looking into their ecology, so that we can make some kind of use of it, just as what I mentioned about the sustainable apiculture. And so, as these arachnids, um, double quotes, they have been called as neglected, but they are now coming into the limelight because they have these kind of important uh, what to say use case. And so curiosity plus careful records is always a kind of research method that everyone can inculcate, so that we can just record new species and then understand their behavior or understand their ecology. And hence, we can try to make these overlooked and neglected arachnids more seen to the general public. So this is a very brief talk that I wanted to give you about my research and my understanding about pseudoscorpions and other neglected arachnids. And I would like to thank my institutions and my funding agencies and my the Biorix community for inviting me, for giving this space to share my thoughts, and the Forest Department for all the support that they had provided during my research. Yeah, so that's all. Thank you.
Do we have any doubts or questions?
Hello, Jithin. Am I audible?
Yes, sir, you are audible.
Jithin, thank you for that very solid presentation. Thank you very much. I have a couple of questions. One is, how do you compare the diversity of pseudoscorpions across the forest and non-forest areas?
So there have not been many studies that have compared the diversity across forest or non-forest areas like plantations or like reserve forest areas. But of course, we can compare them with different. Um, so pseudoscorpions usually they occupy two different habitats. Like one, we can see them on the leaf litter or under the rocks, which are usually dispersal restricted. And another category of pseudoscorpions that is more prone to dispersal, that is they are able to hitchhike on other organisms. They are found on the uh, bark of the trees. So there are different sampling methods that we can employ. Either strictly follow methods that is focusing on leaf litter sampling, like pitfall trapping or collecting leaf litter, and then we get a specific category of pseudoscorpions that is dispersal restricted, and they show micro endemism because mostly they are having short range distribution, so their diversity would be higher compared to that of the bark dwelling pseudoscorpions, which are comparatively more prone to dispersal. So they can actually distribute from one place to another, and hence the species home range is actually greater for those kind of groups. So yeah, if you look into the forest and non-forest areas, it depends on how you sample the regions and what category of pseudoscorpions you are focusing on. The diversity changes. Maybe it is the same species you get across different bark of trees, but it might be different species that you might get from the same leaf litter.
You told that the Parakat gap is not a absolute barrier for Pseudoscorpions. So I suspect that the non-forest areas uh, in the gap is also livable for
Yes,
Pseudoscorpions,
of
right?
course. So non-forested areas like as I mentioned, like many disturbed forests or plantations, or even our backyard can host many pseudoscorpions. But the diversity of those pseudoscorpions that we find in such kind of disturbed habitats or non-forested regions are very less compared to that of pristine forests, because these pristine forests, because these organisms are very sensitive to habitat uh, disturbances. So only those can tolerate such kind of extreme changes in environmental conditions. Um, show this kind of presence in non-forested areas. I mean, that's the observation that I have obtained. Maybe that's a different opinion, but that's what I want to tell you.
My last question, is there any study you have come across where somebody has studied the impact of forest fire on Pseudoscorpions?
Not on pseudoscorpions, but I think there have been studies on spiders, but I am not sure of the details of that, but not on pseudoscorpions. There have not been any studies that I have come across.
Okay. Thank you, Jithin.
Thank
Thank you.
you.
Hello?
Yes, yes.
Hello? The bioluminescence properties are there in most of the scorpions?
So they have this. Scorpions have an integument that responds to UV light, but these are not seen in pseudoscorpions. So pseudoscorpions don't exhibit any kind of bioluminescence or fluorescence. Also,
Thank you.
they don't.
Thank you.
They also scorpions they don't exhibit bioluminescence. They just fluoresce under the UV light. So the pseudoscorpions don't show them.
It's for attracting the partners or something other main work is there for say some other important work is there for them?
There have been that's debatable, but I'm not sure of the correct answer. So there are multiple reasons. I don't know.
Okay. Thank you. Thanks,
Thank
sir.
you.
Thanks, sir. Thank you very much.
Hi, sir. Great talk. I had just one doubt. So you mentioned that pseudoscorpions are like at this point reached to something called as evolutionary stasis, right? Like the lineage is like very less changes in their morphology. So I was wondering why exactly is that happening, and secondly, if it's because they have attained a perfection in their evolutionary race, then can we expect that in next upcoming, like let's say another million years, they'll still look the same, just like that?
Yes, that is something very interesting to look into. Like because these animals have kind of perfected their body plans so that they don't have to change their morphological features according to their environment where they live in. Instead, they just change their ecological niche. Like for example, certain pseudoscorpions they live under the leaf in leaf litter. Some of them live under the tree bark, or some of them even if they live in the desert, they adapt themselves so that or live in such a microhabitats so that they get some kind of moisture. For example, in the deserts, they can be seen under rocks or seen under um, tree bark, or even some of the species, they have slightly modified their integument to have a waxy cuticle that reflects sunlight. So there are some minor modifications that helps them to adapt to ecological niches, but on the whole, their body plan remains the same for a long period of time. So I think they will remain the same um looking forward also. I mean, that's what I understand.
So you're saying that majorly the body plan might be same, but I was thinking like since you mentioned a very minor thing, like let's say the cuticle and how it works. So if they are like adapting to specific niches, wouldn't they be doing have their own evolutionary way? Like let's say one is being adapted to a tree, so their entire lineage is adapted to live on a tree, things like that. Just like the way how birds do, their beaks get evolved accordingly, things like that.
Yeah, actually, we don't see any kind of such kind of uh, very apparent striking differences in their morphology if it is living on the tree or if it is having a phoretic nature that is capable of dispersing from one place to another, or if it is living in the leaf litter where it is not capable of dispersing and it is very endemic to that region. So all these different ecological niches, they kind of represent the same body plan with not major modifications. So that is something very interesting because they try to conserve their morphology and they have been doing this for millions of years. Yeah.
Thank you, sir. One last question. So if I come across a pseudoscorpion, how should I photograph it? Like in order to understand and study about it, in what angles and all.
So as I mentioned, most of the Pseudoscorpions, like the average size comes to about 2 to 3 millimeters, and this is the Pseudoscorpions that you find in the bark of the tree, whereas the leaf litter species are usually coming in the average size of less than 1 millimeter. So you need a good macro setup for getting a live photograph, and usually you take the dorsal and the ventral photograph, and then especially the chelicera, that's the mouth part. So there are certain minute hairs that you can we usually look under the microscope for seeing the different kind of arrangement of the hairs and also the arrangement of the hairs on the chelate pedipalps. That's very important in the case of as far as taxonomy is concerned. So if you just want to take a habitus photo, just take a dorsal and a ventral photo, but that just becomes a habitus photo. For detailed taxonomic investigation, you need a microscope. A good stereo microscope would do the job.
Okay, thank you.
Thank you.
Sir, I think we have a question in the chat box also.
Okay, let me check. Okay, how does Eastern Ghats diversity compare to Western Ghats? Are the dispersal events seen in Eastern Ghats too? I would like to answer that, but unfortunately, I have no data from Eastern Ghats. No one has been working on the pseudoscorpions of Eastern Ghats so far. So, but as the topology of Eastern Ghats is concerned, it's a more discontinuous mountain range compared to that of the Western Ghats. So we expect to see many patchy distributions of different lineages, which might be isolated from one another as compared to the Western Ghats. Because in Western Ghats, the mountains and the forests are more or less continuous, so that there is always a possibility of gene flow and exchange. Whereas in Eastern Ghats, there is more kind of isolation. So allopatric speciation is more expected in Eastern Ghats, but I have no data to convince you that.
Thank you, sir. Any more questions?
Hello, Dr. Jithin. Can you hear
Yes,
me?
yes.
Hello, Akash this side. Thank you for the talk, first of all. It's very rare to hear something about arachnids, which are not spiders usually. My question, so basically in between, I was not able to hear because of my internet issue. So first thing I wanted to ask about the mating system of the pseudoscorpion, how the mating system is, how much inbred it is. And as you were telling, there are cryptic species and the morphology is not changing much. The dispersal is low. These all are hinting towards like uh, uh, success of the evolutionary arms race that we talk about. Might it is also limiting the factor. We can say both ways. Either they have are very successful or either they will struggle in the coming time. So I just wanted to ask across the Western Ghats how the population structure of the pseudoscorpions come along. how much mutation rate we see in these pseudoscorpions, if there is any data on
So
it.
I think it's a two-part question. So first thing is regarding the mating courtship thing about the pseudoscorpions. So it's a very basic methodology. Like the male will lay down the spermatophore in a particular surface, and the female sometimes just senses it randomly, comes across the spermatophore, and then gets into her genital opening, and that's how the fertilization happens. In some more advanced or recent families, we have this courtship dance where the male tries to bring in the female over the spermatophore so that she gets in the spermatophore through the genital opening. So it depends upon the different families, but the basic stuff is same. That is, the spermatophore is just laid down in a surface, and the female just comes in and gets it into her genital opening. So that's the mating thing about pseudoscorpions. And coming to the mutation thing that you mentioned, there is a lot of dark diversity or lot of species to be described or discovered in Western Ghats. My sampling was very restricted to southern Western Ghats and central Western Ghats, and across a period of maybe 1 or 2 years, and a very small sampling effort, I was able to describe about 25 new species and two genus. So there is a lot of mutation and a lot of speciation happening, which we need to discover among these dispersal restricted arthropods. And this is not just about pseudoscorpions, it's about all the dispersal restricted arthropods or invertebrates in common.
Yeah, thank you so much. So the first part of the question, which is the mating system, thank you for describing that. But I wanted to ask more on the side how much inbred the population usually is, because they are less dispersed, as we are talking about again and again. I'm also working on a model system social spiders, which are not as much as like as well dispersed as we talk about other insects or arachnids in general. So I just wanted to ask how much inbred the population. Is there any data on that, or your study suggests something
There
on
might
that?
be some data on inbreeding and introgression and hybridization, like the such kind of population structural study that happens. But from India, or like I have not done any kind of population structure analysis, so I am not able to answer. Yes, sorry.
It's okay. Thank you so much. Thank you so much.
Hello? Hello?
Yes.
I don't know if I missed this part, but what kind of venom do they discharge, scorpions?
Sorry, what kind of venom?
Do they discharge when they strike somebody, as neurotoxic or hemotoxic, whatever it is?
So there is only two papers that have been described different proteins, like cocktail of proteins, and these have suggested certain neurotoxic as well as chemotoxic, cytotoxic, and anti-inflammatory, antibacterial, antifungal properties. So there are a lot different kind of properties that these venom come under, but more details on that I'm not able to tell. Yeah, sorry.
Yeah. Thank you. Thank you. Thank you. Thank you.
Hello, sir.
Yes.
Hello. Thank you so much for this session. I just wanted to ask one simple question. What would be the best time to observe the spider species, like the spider diversity? Is it in the early morning, or what do you prefer, like, or the late night, or what would
Well,
be the best time?
of course, most of these arachnids are nocturnal, so these are mostly active during the night time. So sampling during the night time would yield a much more efficient sample numbers. But for our convenience, because night sampling is difficult, we try to do the sampling in early day hours, not when the scorching sun is out there, because most of the pseudoscorpions they try to hide, or they try to avoid the sunlight or try to avoid the scorching sun, and they try to hide inside the tree hollows or the tree bark, or go under the soil, sometimes not only under the leaf litter but also under the soil, under the top layer of the soil. So it's better that we sample the specimens in the early hours of the day when there's not much sunlight hitting the ground directly. So this is the case for, I think, for most of the soil dwelling arthropods. Yeah, I hope that answers your question.
Okay, thank you, sir. And one more thing, will we get this PPT or the slides which you have shared,
I think
uh,
this
or
right.
any material
I
regarding?
think this recording of the talk is going to be shared with you, right? Um, Gayatri, can you answer that?
Yes, sir. Yes, sir. The YouTube link will be shared later in the group.
Okay, okay, okay. Thank you so much. Thank you so much, sir.
Thank you. Yes.
Hello, Jithin. This is Madan. Your session was very nice. It was very intriguing. Like I have observed one pseudoscorpion in my home in Chennai. So I don't know where I get it from. From your session, I come to know that it usually hitchhike from Western Ghats, right? Most of the diversity is recorded in Western Ghats and Eastern Ghats only. Other than that, any other records we have?
As far as Indian context is concerned, we have more studies from the Himalayan region and from Western Ghats, and also from certain regions of the Eastern Ghats, like from the Madras region, because there were two people from the Madras University who were working on pseudoscorpions, and most of the foreigners work from the Himalayan regions. But the pseudoscorpion that you found in your home, that's kind of a common pseudoscorpion that hitchhike, and it's called as the book scorpion or moss scorpion, and that's a cosmopolitan distributed pseudoscorpion called as the Califer cancroides species. So it can be seen
Oh,
in old libraries. It feeds on book lice and small beetle larvae, and so you can see them in old houses, like stuff like that. So those are kind of synanthropic and cosmopolitan.
thank you, Jithin. Yeah.
So
Good evening, Jithin sir. Hello,
yes.
good evening, Jithin sir. This side Dr. Avinash Perida. Yeah, very nice presentation, Perida. My question is, which gene is expressing spider for like producing silk?
Sorry,
Which genes are expressing?
uh, can you repeat the
Which
question?
genes are which genes are expressing during the silk production?
Oh, which genes are
Which genes is
expressing?
responsible?
Which genes are
Yes.
expressing for spiders, or in the case of pseudoscorpions? I mean, in either cases, I
No,
don't know
no,
which
spiders.
spiders. No, I don't know about
Spider.
spiders. No, I'm sorry, I don't know about spiders.
And scorpion, that venom gland actually in snake that modifies salivary gland. In case of scorpion, the venom gland actually what filter modification, integumentary or some other thing, tail modification.
In the case
The
of
venom gland,
yeah,
the venom gland in case of the snake, it's a modified salivary gland. Okay, so in case of scorpion, what is that? Is modification of tail integumentary system or other
in
thing,
the
a
in
special
the
type of
case of
gland.
scorpion, it is the last segment of the elongated metasoma. That is the last segment of the abdomen that has been modified into the venom apparatus. But in the case of pseudoscorpion, it is the uh, end of the pedipalpal teeth. So the pedipalp has different teeth, and the last teeth has been modified into the venom apparatus in the case of pseudoscorpions. So though that particular teeth ejects the venom in the case of pseudoscorpions. So I think that's the modification that you're looking into.
Okay. So in the integumentary system, how the bile sense which protein or gene is expressing, which is what is the reason behind it? And
So
you will like how this.
Usually pseudoscorpions, they don't actually fluorescent under the UV light, but scorpions do. But I am not sure what proteins are expressed in the integument of scorpions. No, I'm not sure about scorpions because my primary interest is in pseudoscorpions, so I'm not an expert of scorpions. Sorry.
Okay, okay, fine. No issue. Okay, fine. Thank you.
I suppose there are many other questions.
Okay, so I think all your doubts have been cleared. So, as we come to the end of today's session, we extend our sincere thanks to Dr. Jithin Johnson for delivering an insightful and engaging talk on the less talked about side of arachnids. We also thank everyone who joined and actively participated. So I would also like to remind you all about Biorix Biotalks. It is a student knowledge sharing series, which is a wonderful platform for you to present your insights on everything biology related. Registrations are still open. So if you're passionate about any topic in biology or life sciences, this is your opportunity to step up and lead a session with Biorix. Interested participants can register through the form shared in the Biorix group or through the link in our Instagram bio. So if you would like to see a bio talk session, tomorrow we have a Biorix Biotalk session titled Nature's Incognito Mode, the Evolutionary Mastery of Disguise, Exploring Mimicry and Coloration in the Natural World. So it is by Miss A. Bharati, a third year student of BSc Zoology, Justice Bashir Ahmed Saeed College for Women, Chennai. So we hope all of you will join us tomorrow and also try to register for the Biotalks program. So, with that, we officially conclude today's session. Thank you all for participating in this discussion. See you in
Thank
the
you.
next Biotics session.
Okay, thank you. Bye bye. Good night.